Welcome to the PubMed Daily Review for April 06 SD. Today we scanned 1389 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Biomaterials, Digestion, and Microbial Ecology. Investigating how biological materials interact with and influence microbial ecosystems and digestive processes. This cluster covers 275 papers. Representative work includes zirconia and titanium implants placed at 2 supracrestal levels in fresh extraction socket. Canine periodontitis model. Standardized ileal digestibility of amino acids in L-amino acid sources and single-cell protein biomass coproduct for pigs, and multi-omic insights in a Mediterranean diet-associated microbiota. Trend 2 of 5, Liver disease prediction and AI. AI and advanced diagnostics are revolutionizing prediction and management of diverse medical conditions. This cluster covers 351 papers. Representative work includes non-invasive tests for predicting decompensation in compensated advanced chronic liver disease, a comprehensive review, echocardiography, bile acids and spleen stiffness to rule out high-risk varices in children with biliary atresia, and impact of disease-specific treatment and non-selective beta blockers on risk of PVT in cirrhotic patients with HCV or PBC. Trend 3 of 5, Healthcare training, public health, and social issues. This trend explores health interventions, patient well-being, and societal factors impacting public health outcomes. This cluster covers 332 papers. Representative work includes partnerships in learning, the Rural Health Multidisciplinary Training Program in Aged Care Services Pilot Site Evaluation, clinical and epidemiological factors associated with severe dehydration from cholera among pediatric patients in the community of Lusaka, Zambia, October 2023 to May 2024, and indirect mobilization and violence legitimation through influencers on alternative platforms. Trend 4 of 5, Neuroscience, Cancer, and Women's Health. Investigating neurological mechanisms and treatments across diverse conditions, including cancer, injury, and women's health. This cluster covers 161 papers. Representative work includes glia or bussin, how single-cell and spatial transcriptomics enlighten the role of neuroglia in spinal cord injury and regeneration, direct optimization of the probability of lesion origin in proton treatment planning for low-grade glioma patients, and a novel transcutaneous auricular neurostimulation electrode configuration for treatment of heavy menstrual bleeding, an open-label trial. Trend 5 of 5, Regenerative medicine and disease models. These papers explore how biological processes and models can advance disease understanding and therapeutic development. This cluster covers 270 papers. Representative work includes murine progeria model exhibits delayed fracture healing with senescent phenotype and dysregulated immune response. B-cell differentiation of human hematopoietic progenitors is efficiently supported by Wharton jelly-derived mesenchymal stem cells, and RXR-gamma enables oligodendrocyte differentiation by suppressing sonic hedgehog signaling. Up next, our first paper out of 5, Protective gene signatures in the transition from SIRS to sepsis. Insights from Integrative Transcriptomics and Validation across clinical cohorts by Qingmin Gu and Feng Yang from Department of Emergency Medicine, the Affiliated Hospital, Southwest Medical University, Luzhou 646000, People's Republic of China. Let's break down its key contributions. Sepsis remains a major clinical challenge with a high mortality rate. It often develops from systemic inflammatory response syndrome, or SIRS. However, the precise molecular changes that drive this progression from SIRS to sepsis are not well understood. Current clinical markers often overlap between these two states, making early differentiation difficult. This study aims to uncover the key protective genes involved in this critical transition, offering potential new avenues for diagnosis and treatment. This study employed a multi-pronged approach. They analyzed RNA sequencing data from patients with sepsis and SIRS, alongside publicly available datasets for validation. Advanced computational methods like WGCNA and machine learning were used to pinpoint crucial genes. Furthermore, single-cell RNA sequencing and scenic analysis provided insights into cell-specific gene expression and regulatory networks. In vitro experiments with macrophages further validated the findings. The key finding was the identification of 2 genes, CDSS and NRG1, as potential protective factors. These genes were significantly downregulated in patients with sepsis compared to those with SIRS. This downregulation was consistently observed across multiple independent clinical cohorts, strengthening the findings. Further analysis showed these genes are primarily expressed in immune cells like monocytes and are also reduced in response to inflammatory stimuli in macrophages. In conclusion, this research highlights CTSS and NRG1 as promising biomarkers that could help identify patients progressing from SIRS to sepsis. Their specific expression patterns in immune cells offer new insights into the complex immune dysregulation seen in sepsis. Ultimately, these findings pave the way for exploring CTSS and NRG1 as potential therapeutic targets to mitigate sepsis progression and improve patient outcomes. Up next, our second paper out of 5. IDH1 mutation creates a dependency on fatty acid metabolism that underlies sensitivity to cuproptosis in acute myeloid leukemia cells by Xuening Zhang and Wenjun Ma from State Key Laboratory of Mechanism and Quality of Chinese Medicine and School of Pharmacy. Faculty of Medicine, Macau University of Science and Technology, Macau, SAR 999078, China. Let's break down its key contributions. Acute myeloid leukemia with IDH1 mutations presents a significant clinical challenge. While targeted therapies exist, they don't work for everyone, and resistance is a problem. This study explores a new angle, cuproptosis, a copper-induced cell death pathway, and its potential to treat IDH1 mutant AML. To investigate this, researchers used both AML cell lines with and without IDH1 mutations, as well as mouse models. They treated these models with ilesclamol, a compound that triggers cuproptosis, and analyzed the cellular responses using transcriptomics and metabolic assays. The key was to see if IDH1 mutant cells reacted differently to this treatment. The results were striking. IDH1 mutant AML cells showed significantly greater sensitivity to ilesclamol compared to their wild-type counterparts. Ilesclamol treatment led to a broad downregulation of lipid metabolism pathways. Importantly, when fatty acid availability was restricted, the IDH1 mutant cells became even more vulnerable to ilesclamol, and this translated to reduced tumor growth in vivo. This study uncovers a critical metabolic dependency in IDH1 mutant AML cells, revealing a potential new therapeutic avenue. By exploiting the vulnerability to cuproptosis and fatty acid metabolism disruption, we might develop more effective treatments. Future work should focus on translating these exciting findings into clinical applications. Up next, our third paper out of 5, Beyond Antibiotics, a new antivirulent strategy combating ESBL-producing Klebsiella pneumoniae by targeting paraplasmic trihalase by Qinhua Zhang and Yin Wang from Respiratory and Critical Care Medicine. The Second Hospital of Jilin University, Changchun, Jilin 130041, People's Republic of China. Let's break down its key contributions. The rise of antibiotic-resistant bacteria like ESBL-producing Klebsiella pneumoniae is a major global health concern. These infections are difficult to treat and have high mortality rates. While antibiotics are the current standard, their overuse drives further resistance. This has led researchers to explore novel strategies, such as antivirulence therapies, which aim to disarm bacteria by targeting their ability to cause disease, rather than killing them directly. This approach could potentially reduce the selection pressure for resistance. To investigate the role of paraplasmic trihalase, or TRI, researchers created a version of ESBL-KP that couldn't produce it, known as a trait-deficient mutant. They then compared this mutant to the original wild-type strain. This comparison involved assessing several key aspects: how well the bacteria grew, their ability to survive heat stress, how they fared inside immune cells called macrophages, their capacity to form biofilms, and ultimately, their virulence in a mouse model of lung infection. The goal was to pinpoint the specific contribution of TRI to the bacteria's ability to cause disease. The results were quite striking. The bacteria lacking TRI showed significantly slower growth and produced less biomass in the stationary phase. They were also less able to tolerate high temperatures, with the mutant strain exhibiting reduced survival under heat stress. Furthermore, when tested inside macrophages, the trait-deficient bacteria survived less effectively. Biofilm formation was also notably reduced. Most importantly, in the mouse lung infection model, the TRI mutant displayed significantly lower virulence, leading to better survival rates for the infected animals. These findings strongly indicate that tree plays a crucial role in the pathogen's ability to cause infection. So, what does this mean for the clinic? This study identifies paraplasmic trihalase, TRE, as a key player in the virulence of dangerous ESBL-producing Klebsiella pneumoniae. This discovery opens the door for developing new antivirulent strategies that specifically target TRE. Such an approach could provide a much-needed alternative to conventional antibiotics, helping us combat these increasingly resistant superbugs. While promising, further research is essential to translate these findings into effective and safe treatments for patients. Up next, our fourth paper out of 5, VP, toward a new generation of error-free prime editing by Monur Jamil and Sultan Habibullah Khan from Center for Advanced Studies in Agriculture and Food Security, University of Agriculture, Faisalabad, Pakistan. Let's break down its key contributions. Prime editing is a revolutionary tool for making precise changes to our DNA. Think of it like a search and replace function for the genome. However, current prime editors aren't perfect. They can leave behind unedited DNA strands and often introduce unwanted errors, called indels. These issues limit how effectively we can use this technology for treating diseases or improving crops. To overcome these challenges, researchers engineered a new prime editor, which they call VP, or Very Precise P. The key innovation lies in introducing specific nick-relaxing mutations into the editor. This design aims to destabilize the unedited 5' DNA strand and promote degradation of the nick strand. The goal is to significantly reduce those problematic indel errors and make the desired edits more stable. The results are very promising. This new VP system successfully minimizes the rate of indel errors that plagued earlier prime editors. Furthermore, it actively favors the incorporation of stable, intended edits into the genome. This breakthrough suggests VP could be a transformative tool for developing new therapies for human diseases and driving innovation in agriculture. So what does this mean? The development of VP marks a crucial step forward in making prime editing a more reliable and error-free technology. By minimizing unwanted DNA errors, VP holds immense potential for developing safer and more effective gene therapies. While more research is needed to fully realize its potential in both medicine and agriculture, this is a very exciting development for the future of genome editing. Up next, our fifth paper out of 5, a single-cell and spatial transcriptomic atlas of human tuberculous constrictive pericardium by Feng Xiong and Yi Wang from A Department of Cardiology, Affiliated Hospital of Southwest Jiaotong University, the Third People's Hospital of Chengdu, 610031, Sichuan, China. Let's break down its key contributions. Tuberculous pericarditis can lead to a dangerous thickening and scarring of the pericardium, known as constrictive pericarditis. This condition severely impacts heart function. While we know inflammation and fibrosis are key, the precise cellular players and how they interact to cause this scarring have been a mystery. This study aimed to unravel these complex mechanisms at a single-cell level. To investigate this, researchers analyzed pericardial tissue from patients with tuberculous constrictive pericarditis and compared it to healthy controls. They employed advanced techniques like single-cell RNA sequencing to understand individual cell types and spatial transcriptomics to see how these cells are organized within the tissue. This allowed them to map out the cellular landscape and identify key signaling pathways involved in the fibrotic process. The study revealed a complex network of cells within the diseased pericardium. They identified 14 distinct cell types, highlighting specific macrophages, endothelial cells, and fibroblasts as key players. Crucially, they found that macrophages release signals like VEGF and TGF-beta, which drive endothelial cells toward the fibrotic state and activate fibroblasts. This intricate cellular crosstalk appears to be the engine behind the excessive scarring seen in TBCP. This research provides a groundbreaking map of the cellular and molecular events driving constrictive pericarditis and tuberculosis. It shows that the fibrosis is an active, orchestrated process. By pinpointing key cell types and signaling pathways, this work opens doors for developing new targeted therapies and biomarkers to better manage patients with this severe condition.